Assalamualaikum, lovely students. Today we'll be looking at acid-fast staining. But before getting into the video, I'd like to tell you guys that these videos are meant for educational purposes. Things and treatments may change with time. If I get wrong or miss anything, your input is always welcomed in the comments section. Have a cup of tea and let's get started. Acid-fast stain is a differential stain that is used to identify and detect acid-fast organisms. Who are the acid-fast organisms? We'll be looking at them in just a few moments. The example of acid-fast organisms are Mycobacterium tuberculosis, that is responsible for causing TB, the tuberculosis. That's a strict acid-fast bacterium. While the Nocardia asteroides, I do have a video on that, is weakly acid-fast organism. Acid-fast bacteria appear red on acid-fast staining, while the non-acid-fast bacteria appear blue on acid-fast staining. As you can see in this picture, these are the acid-fast bacteria and they are red colored. And these ones are the non-acid-fast bacteria and they stained blue on acid-fast staining. Lecture outline. As we are done with the introduction of acid-fast staining, now we'll be looking at the acid-fast organisms, reagents and equipment required to perform the acid-fast stain, then what's its procedure, what are its different types, and at the end, as usual, we'll review the lecture. Acid-fast organisms are those who stain red on acid-fast staining. They've got wax-like, almost impermeable cell walls. This sign is for approximately. And they contain mycolic acid, fatty acid, waxes, and complex lipids in their cell wall, and these things make the cell wall of acid-fast organisms resistant, which means that it's impermeable to certain substances. And the acid-fast organisms do not stain with other stains, for example, gram staining. But there are certain exceptions, like for Nocardia asteroides, as it's a weakly acid-fast bacterium, it also stains with gram staining, and it's a weakly gram-positive bacteria. But for Mycobacterium tuberculosis, it does not stain with gram staining. Acid-fast organisms are resistant to disinfectants, dry conditions, and other stains. I've got this picture as a visual mnemonic for you. Like acid-fast organisms are resistant to disinfectant, dry conditions, and other stains. Reagents required. The first thing that we'll need is carbonyl fusion. That's a primary stain. Then we'll need methylene blue. That's a counter stain. Then we'll need acid alcohol. That's a decolorizing agent. And we'll also need distilled water. And we'll definitely need certain equipment like burner, dropper, etc. Procedure. Prior to talking about the steps, we'll do what? We'll go for smearing of the specimen on the slide. We'll take the specimen from this test tube and we'll smear it on the slide. Now let's talk about the steps. There are a total of 4 steps that are involved in acid-fast staining. First one is addition of carbonyl fusion. That's the primary stain. Then heat fixation. The third step is decolorization, that is done with acid alcohol. And the last step is counter staining, that is done with methylene blue. That's the counter stain. Let's talk about each step in detail. Step 1, the addition of carbonyl fusion. But prior to adding, we should know what actually is a carbonyl fusion. Carbonyl fusion is a lipid-soluble compound and it contains phenol in it. And these two things does what? They help the stain to penetrate the cell walls, as the cell walls are resistant, impermeable, and wax-like. Carbonyl fusion is composed of basic fusion, ethanol, heat-melted crystals of phenol, and distilled water. As you can see, this is a carbonyl fusion. It will does what? It will stain all the bacteria red, whether they are acid-fast or non-acid-fast, and it will be incubated for 30 seconds, and after 30 seconds all the bacteria will be red, as you can see there on the slide. Step 2, heat fixing or fixation. After addition of the carbonyl fusion, we'll take the slide and we'll heat it up. That will fix the stain. This helps the bacteria to stick to the slide and bacteria will not wash out with other reagents. Third step is decolorization. It means to add a decolorizer. Now, after fixing that bacteria on the slide, rinse it with a very strong decolorizer. We've got acid alcohol, that's a strong decolorizer. We'll add its drop, as you can see there. This decolorizer strips the stain from all non-acid fast bacteria. All the bacteria were red as we added carbofusion, but when we'll add the decolorizer, it will take away the color from non-acid fast bacteria, as you can see there. And the decolorizer does not penetrate the cell wall of acid-fast bacteria, so it cannot wipe out the stain from acid-fast bacteria and they will remain red, just like that. The decolorizer used is very strong and it contains ethanol and hydrochloric acid, and that's the concentrated one. Last step, the counter staining. We'll add the methylene blue that acts as a counter stain. The decolorized non-acid fast bacteria will stain blue with the counter stain, as you can see in this picture. Types of acid fast stain: Zeil-Nielsen method and Quignon method. These are two different types of acid fast staining. There's one another, but we'll talk about that in just a moment. Zeil-Nielsen method is most common one. It uses heat to enhance the penetration of carbofusion. But on the other hand, Quignon is the modification of Zeil-Nielsen stain and it does not require heat. Carbofusion used in Quignon method is more concentrated and it contains a wetting agent that facilitates the staining process. In Zeil-Nielsen stain, after the penetration of carbofusion, the smear is then decolorized with acid alcohol, and then it is counterstained with methylene blue, and acid fast bacteria retain the red color and they appear red, while the non-acid fast bacteria are decolorized and then they get the color from methylene blue and they appear blue. Decolorization and counterstaining steps of Quignon are similar to those of Zeil-Nielsen's steps. As you can see in this picture, the acid fast bacterium is red, while the non-acid fast bacteria are blue. The other dye is auramine-rhodamine stain. It's a fluorochrome stain. Instead of carbofusion, fluorescent dyes are used in this staining method. These dyes bind to the mycolic acid in the bacterial cell wall. Smear is then decolorized with acid alcohol, and after that, it's counterstained with what? Potassium permanganate, not with methylene blue, as was done in normal acid-fast staining. The acid-fast bacteria will emit a yellow-green fluorescence, and non-acid-fast bacteria appear dark or quenched. As you can see in this picture, these bacteria emit a fluorescence, but here you cannot see clearly which color is that, but actually it's yellow-green color. Alright guys, let's review everything really quick. We discussed acid-fast staining today. We use carbofusion as a primary stain to stain all the bacteria, whether they're acid-fast or non-acid fast, red. After that, we go for decolorization and we use a decolorizer that is acid alcohol, and that wipes out the stain from non-acid fast bacteria, because acid fast bacteria has got a resistant cell wall and acid alcohol is not able to wash out dye from its wall. So non-acid fast bacteria will appear just like that, and then we'll use a counter stain in acid fast staining that is methylene blue. We'll use it will color all the non-acid fast bacteria blue, and acid fast bacteria are still red. And that's it for today's video. I hope you enjoyed it. If you've got any suggestions, feel free to leave them below in the comments. And also, if you want to connect with me on my socials, I've got my Instagram, Twitter, and I'll catch you in the next video. Till then, Assalamualaikum.